Hi everyone, this is Dr. Ross, and this is going to be a video presentation going over the anatomy of the lymphatic system as well as a little bit of histology. So I'm going to start with the primary lymphoid organs. These are the organs where lymphocytes mature, proliferate, and are selected for. Selection just means it enables them to attack pathogens without harming the cells of the body. So this is where the ones that would recognize self should be removed from circulation. So the two primary lymphoid organs are the bone marrow, shown in this image, and the thymus, which I'll get to in a second. So the red bone marrow, specifically, is a loose collection of cells where hematopoiesis occurs. Recall, hematopoiesis is the formation of red blood cells, white blood cells, and platelets. Both B and T cells are made here as well. So B cells mature in this bone marrow, and that's why they're called B cells. T cells are the other type of specific immune cell. They are formed in the bone marrow, but they complete their development in the thymus. That's why they're called the T cells. When T cells leave the bone marrow, they're actually called a thymocyte. When they finish their development, we will call them a T cell. So the thymus gland, shown here, is a bilobed organ, so two lobes found in the space between the sternum and the aorta of the heart. Connective tissue holds the lobes closely together, but it also separates the two lobes and it forms the capsule. So on the top image here on the right, you're seeing some histology of the thymus, and the bottom is obviously a cartoon representing these same features. So on the outside, we see here this fibrous capsule that I've just highlighted. Now that connective tissue capsule further divides the thymus into lobules via extensions called trabeculae. And you can see those kind of, the capsule or that connective tissue kind of diving into the thymus, and that's those trabeculae. The outer region of the thymus, below the capsule that is the cortex. Okay, so the cortex contains large numbers of thymocytes. Remember, thymocytes are the cells that have left the bone marrow, but they're not completely mature. There's also epithelial cells here, macrophages, and dendritic cells. Dendritic cells are phagocytes, meaning that they're going to engulf. So are macrophages, they're also phagocytes. They're going to engulf foreign materials. The cortex is densely packed, so it stains more intensely than the rest of the thymus, and that can help you identify it. The inner part of the thymus is called the medulla. This is where the thymocytes migrate before leaving the thymus. This contains a less dense collection of thymocytes, epithelial cells, and dendritic cells. Now I want you to note that the thymus gland begins shrinking at birth. This happens at about a rate of 3% per year, and it continues at this rate until you're about 35 to 45. Then that rate will decline to about 1% per year for the rest of your life. I've read somewhere that it's estimated that the thymus would completely disappear if we live to age 120. But the shrinking of the thymus probably has a significant amount of influence over how our immune system becomes less effective as we age. All right, so secondary lymphoid organs are where the lymphocytes mount an immune response. Okay, so this is where the lymphocytes, the T cells and B cells, do their job. So they're going to encounter pathogens here, they're going to encounter toxins, as well as other non-self particles. So let's start with the spleen, which I've shown here. This is a major secondary lymphoid organ. It's sometimes called the filter of the blood because of its extensive vascularization and the presence of macrophages and dendritic cells that remove microbes and other material from the blood, including the dying red blood cells. The spleen also functions as the location of immune responses to blood-borne pathogen. The spleen contains several histological features, including the outer capsule, which is shown here in this histological micrograph. You can also see the red pulp, which consists of reticular fibers with fixed macrophages attached. Free macrophages and all the other cell types of the blood, including some lymphocytes as well, are going to help function in this filtration. You can also see white pulp. The white pulp surrounds a central arteriole and consists of germinal centers. Germinal centers are where you have dividing B cells surrounded by T cells and accessory cells. That's going to include the macrophages and dendritic cells as well. The white pulp is where the specific immune response is mounted. All right. Now, next we have the vermiform appendix, or the appendix. This is a tube that's connected to the cecum. The appendix has been identified as an important component of the mammalian mucosal immune function. So a lot of people used to think the appendix was just a vestigial organ, but it seems to play a very important part in immunity. It's particularly important for B cell mediated immune responses and extra thymically derived T cells, so things that are derived outside the thymus. In 2007, it was proposed that the appendix also serves as a haven for useful bacteria. When illness flushes the bacteria from the rest of the intestines, they think the appendix may act as a safe house for beneficial bacteria that then kind of repopulate the intestine. So lymph nodes also function to remove debris and pathogens from the lymph, and thus are sometimes referred to as filters of lymph. Any bacteria that infect the interstitial fluid are taken up by the lymphatic capillaries, and then they're transported to the regional lymph node. Dendritic cells and macrophages within this organ internalize and kill many of the pathogens that pass through, and that's how a lot of organisms are removed from the body. Like the thymus, the lymph nodes are surrounded by a tough capsule. And so again, at the top, you can see a drawing of the thymus, and then at the bottom there is a micrograph that's been stained, so that is our histology. A micrograph of tissue that has been stained. Okay, so like the thymus is surrounded by that capsule, it's connective tissue, and also it's separated into compartments by trabeculae. You can see these in each of the images. It's a little more difficult to see the ones in the bottom image. The major routes into the lymph nodes are the afferent lymphatic vessels. You can see those here at the bottom of the top image. And then within the cortex of the lymph nodes are the lymph node follicles. And this consists of the germinal centers of rapidly dividing B cells. And again, those are also going to be surrounded by a layer of T cells and other accessory cells. As the lymph continues to flow through the node, it enters the medulla, which I've just indicated by an M here. And this consists of medullary cords of B cells and plasma cells. The medullary sinuses, where the lymph collects before leaving the lymph node, will eventually drain into the efferent lymphatic vessels, which are shown in the top image. All right, so we need to know several of the lymph nodes that are present throughout the body. So lymph nodes are more concentrated near and within the trunk, and they are divided into groups. So I'm going to go through the lymph nodes that I want you to be familiar with. Luckily, these mostly carry the name of the body regions, so that should be helpful for those of you that learned your body regions in Anatomy and Physiology 1. So before I do that, I want to mention that some lymph nodes can be felt when they are enlarged, and sometimes when not. And this is going to be, I want you to note this when I talk about the axillary lymph nodes under the arm and the cervical lymph nodes of the head and neck, as well as the inguinal lymph nodes near the groin. So here you can see the cervical lymph nodes located near the head and neck. Here are the axillary lymph nodes in the axillary region or armpit area. Here are the intestinal lymph nodes. I want you to note in this image they are drawn by an intestine, a piece of intestine, the large intestine in this case. You also have the iliac lymph nodes. These are in the abdominal area, but they are not the same as the intestinal lymph nodes. And then you have the inguinal lymph nodes as well. Okay, so as I mentioned, the cervical lymph nodes, the axillary lymph nodes, and the inguinal lymph nodes near that groin crease are nodes that are going to be palpable. All right, so let's move on to the lymphoid nodules, or which includes tonsils. So these have a simpler architecture than the spleen and lymph nodes. They consist basically of just a dense cluster of lymphocytes. They typically don't have a surrounding fibrous capsule. The nodules are located, these types of nodules are located in the respiratory and digestive tract areas that are exposed to environmental pathogens. So things that are exposed to outside. So first, let's start with the pharyngeal tonsil. This is also referred to as the adenoid. It's a massive lymphatic tissue located behind the nasal cavity in the roof of the nasopharynx. This is where the nose kind of blends into the throat. The lingual tonsils are a collection of lymphatic tissues located in the lamina propria of the root of the tongue. And we also have the palatine tonsils. These are commonly just called the tonsils. And they're located in the left and right sides of the back of the throat, which can often be seen as flesh-colored pinkish lumps. The tonsils only present as white lumps if they are inflamed or infected with symptoms of like that have pus drainage or severe swelling. If we take a look at the histology of a tonsil, in this case I'm looking at a palatine tonsil, we can see some specific features. So you see the invagination at the top of the image. Oops, hang on, sorry. The invagination at the top of the image is a crypt. The crypts are there because, one, they greatly increase the surface area that allows contact between the environment and the tissue without increasing the overall size. Macrophages and other white blood cells are going to concentrate by these crypts, and they are also excuse me, hang on, I lost my train of thought. Okay, so they also serve as early alerts for the immune system. So you get exposure to immune cells here, and then that's going to kind of alert the immune system. Unfortunately, the crypts also provide a really inviting environment to some bacteria, and so colonies can form here that solidify and cause plugs or stones within the crypts. And then people who have this will often be those who suffer from like chronic sinusitis or postnasal drip. And they're going to have a lot of this bacterial overgrowth in the tonsil or crypts. These little small plugs or stones are called tonsillolis, and they're sometimes known as tonsil stones. They have a really foul smell, and they often will contribute to really terrible bad breath. They can also obstruct the normal flow of pus from the crypts, and they can irritate the throat as well. You can also see in this image some germinal centers. And like the other tissues, the germinal centers are the sites of actively dividing immune cells. All right, so on this slide, we're going to talk about the mucosa-associated lymphoid tissue, or MALT, M-A-L-T. So this is going to consist of an aggregate of lymphoid follicles directly associated with mucous membrane epithelia. In this top left image, you can see this dome-shaped structure that's found underlying the mucosa of the gastrointestinal tract. But you can also find these in the breast tissue, lungs, and eyes. So Peyer's patches, which are shown here, are a type of MALT in the small intestine, and they're really important for immune responses against ingested substances. So recall that when we eat, of course, this is from outside. Our food is not sterile. So there's going to be organisms in there that need to be removed from the body. So Peyer's patches contain specialized endothelial cells called M cells. And what those cells do is they sample that material from the intestinal lumen. The lumen I've just shown here with this yellow area, it's just the inside of the intestine where the poop goes through, okay, or the undigested food. And so they're going to survey what's in there. And then anything that is found is going to be transported to the nearby follicle so that an adaptive immune response to potential pathogens can be mounted. A similar process occurs in the mucosa and submucosa of the appendix. Other structures of note in the mucosa are the villi, which I just pointed at. These are finger-like projections of the mucosa. These increase the surface area of that tissue. That's what they kind of appear like these projections, but what they do is they increase the surface area, which increases the interaction of the intestine with the materials passing through, which of course the main focus of the intestine is to ingest or absorb molecules. So the more surface area you have, the more absorption you're going to take, or you're going to have. And we'll talk a little bit more about that when we get to the digestive section. You can also make out these germinal centers here, and these are easiest to see in the cross section at the bottom of the image. So the germinal sections are shown there, and again, just like the other ones, this is going to be the site of actively dividing immune cells. So now I'm just going to go through the lymphatic vessels that you need to know. So we've got the jugular trunks. You have a left and right jugular trunk. You have the left and right subclavian trunk. You have the lumbar trunk. These are going to receive lymph from the lower limbs and the walls and viscera of the pelvis, as well as the kidneys. Ultimately, these are going to empty into the cisterna chyli, which I'll talk about in a second. You also have the intestinal trunk. This receives lymph from the stomach and intestine, as well as the pancreas and spleen. It's also going to empty into the cisterna chyli. And then here is that cisterna chyli. It's a dilated sac, as you can clearly see, at the lower end of the thoracic duct. Okay, and so that intestinal trunk and the lumbar trunks are going to flow here. The cisterna chyli receives fatty chyle from the intestines, and that is because the fats and lipids from our food cannot really go through the bloodstream. So they have to re-enter through the lymphatics, and so that's going to happen here in this cisterna chyli as one of the early points in that drainage. Next is the thoracic duct. The thoracic duct is the larger of the two lymph ducts of the lymphatic system. It carries chyle, which is the liquid that contains lymph and fats. It also collects most of the lymph from the body. All right, so this is going to drain most of the body lymph back into systemic or blood circulation. And then last we have over here the right lymphatic duct. This is the lymphatic vessel that drains the right upper quadrant of the body. So this is going to drain less of the lymph back into the systemic circulation. And before I leave, even though this was gone over in the lecture slide, I just want to point out here we have that part of the body in darker green. This is what's going to drain into the lymphatic duct, and the light green is what drains into the thoracic duct. And that is it. Thank you.